Hi, welcome to my Zoom video lab for microbiology. This is for lab number 25, and it is the video lab for inoculations that I was actually planning for Wednesday, April 8th. And it turns out that I totally forgot that this week we have Good Friday holiday, which is April 10th. I'm going to do a video lab tomorrow anyway, and you can just watch it online over the weekend. So I wanted to get that to let you know that it will be available. That lab is going to be due next week. And then next week I will do video labs for lab 45 and 46. So anyhow, I first will walk you through the inoculations. What we're doing is we're testing antibiotic discs. And what I have is a large Petri dish that looks like a dessert plate that this has Mueller-Hinton agar in it. And this is a special agar that will grow up a lawn of bacteria. This is going to be Pseudomonas aeruginosa. I also previously inoculated E. coli, as well as Staphylococcus epidermidis. So, and I will do the inoculation in just a minute for Pseudomonas aeruginosa, and then we will put antibiotic discs directly on the Petri dish, and we can do the Kirby Bauer test that we talked about when we talked about antibiotics and antibiotic resistance and susceptibility. So I will go ahead and get set up for the inoculation. I'm going to take liquid culture of Pseudomonas aeruginosa, spread it in a thin layer across the Petri dish, and then we'll put the antibiotic discs on directly. Okay, hopefully you can see that. Okay, let me start my Bunsen burner. Okay. Open up my Petri dish. Actually, I'm going to do this Pac-Man style. So I've got my Pseudomonas aeruginosa culture, and I've got my sterile swab. Okay, this time I'm not going to pour culture all over the bench like I did last time. So we just simply get our swab out, if it will come out. They always get stuck. There it goes. We can replace the tube. Okay. Then I open up the Pseudomonas aeruginosa, flame the top. Sorry, the Bunsen burner is out of the shot, but you can get the idea. Then I'll go ahead and moisten my tip of my swab, and we'll go in one direction, form a confluent layer of culture. Then I will dip in again and do 90 degrees opposite that. Then I will dip in a third time and tilt the Petri about 45 degrees and go on a diagonal. Okay, so I'm done with my inoculation. And now I can add my antibiotic discs directly to the Petri dish. Okay, we need to get rid of this. So I'll set up to do my antibiotic discs next. So now I have forceps. I have ethanol, which I'm going to use to sterilize the tips of my forceps. I have my inoculated Petri dish, now inoculated with Pseudomonas aeruginosa. And then I have my antibiotic discs in these dispensers. I have chloramphenicol, penicillin, bacitracin, tetracycline, and streptomycin. Okay, so I have those 5 different antibiotics. Now what I'm going to do, so you can see this as best possible, I'm going to take the cover completely off of my Petri dish, which normally is a big no-no, but it's what we're going to have to do today. Okay, then I spritz with ethanol my forceps, and I flame them. Okay, then I get my first antibiotic disc out, which is chloramphenicol. These dispensers do not work well, but we're just going to do our best anyway. So you work at pulling your antibiotic disc out of the dispenser. And then you take your antibiotic disc and you place it in an area far away from the edge and far away from any other discs. Squirt with ethanol one more time. Flame. Then we go to our next disc, which is penicillin. Squirt again with ethanol. Then we flame our forceps. Then we move to bacitracin. Whoops. Now, if the bacitracin disc hits the bench, then it's contaminated, so we have to get another one. Now we have tetracycline. And then finally streptomycin. We can replace the top of our Petri dish, and then we'll go ahead and put this in the autoclave, or not the autoclave, but in the incubator until tomorrow. And then tomorrow we'll measure the zones of inhibition. Each one of these antibiotic discs is going to inhibit growth around the disc itself. So we'll measure these zones of inhibition, and then we'll use a special chart in your lab textbook to let us know whether the bacteria is resistant, susceptible, or intermediate to that particular antibiotic. Okay, I'm going to pause right now and do a little house cleaning, and we'll do critical thinking and clinical applications for Lab 23. Okay, so I've got my book right here. We'll do critical thinking and clinical applications for Lab 23. Lab 23 is due on Monday, April 13th. So you can't do it until you get the critical thinking and clinical applications. So make sure that you have previewed this video. So critical thinking number 1, this is page 193. Why are there still some colonies growing in areas exposed to ultraviolet light? Okay, remember, there were some instances, especially with Serratia marcescens, where outside of the control zone where we had the sunglasses, there were bacterial colonies growing. And so what happens is that due to the selection pressure, that is the UV light, due to that selection pressure, then you will get colonies that have either previously mutated to have strong resistance to UV light, or are mutated in situ to have resistance to UV light, and they will survive that. So you have an experience in natural selection going on, okay? And number 2, how might the results differ if a UVA lamp were used? Now remember, the UV light that we were using is UVC. UVC has a smaller wavelength and is much more penetrating than UVA or UVB. The mildest form of light is UVA. So you wouldn't expect to have a whole lot of damage to the cells or to the cellular DNA if you used a UVA lamp. That is the mildest form of UV light. UVB is intermediate. It's an intermediate form of light, so you would expect an intermediate amount of damage. And of course, we had the highest amount of damage when we were using our UV radiation, which was UVC. Okay, and then number 3, considering your results, discuss the possible effects of UV radiation on the ecology of a lake if the UV radiation had the same effect on lake bacteria as it did on bacteria in your experiment. Well, we saw from our experiment that the UV radiation destroyed the bacteria. It had a very hard time growing. So if we destroy the bacteria in a lake, this goes back to chapter 24 in your textbook, then that is going to destroy the lowest portion of the food web, and the rest of the food web would merely starve to death. So in this instance, you could wipe out the entire biome or entire ecology of the lake if UV radiation were allowed to destroy all the bacteria that smaller animals were feeding on, that small fish would feed on, that large fish would feed on, and eventually that would work its way through the food web and the lake would not be able to sustain life. Okay, on clinical application, you have a table. The table is based on bacterial counts of Legionella pneumophila. This is a type of bacteria that's linked with Legionnaire's disease. And in this table, they have tabulated the amount of bacteria that you would see under control conditions at 23 degrees Celsius, under high heat sterilization conditions at 50 degrees Celsius, under the use of ozone at 23 degrees Celsius, and then under the use of UV light at 23 degrees Celsius. And then each treatment method is then timed for 0 minutes, 20, 40, 80, 160, and 320 minutes. And what you need to do here with this data is you need to fill in the blanks of what you would think would happen if these trends continued for 50 degrees Celsius at 320 minutes, for ozone at 320 minutes, and then for UV light at 320 minutes. So you just want to follow the same trends that you're seeing as time passes in each one of these sterilization methods. And then the question asks, which is the most effective method? And that the most effective method would be the one that would have the lowest count of bacteria after 320 minutes. And then it also asks, there are 2 questions that you want to answer below the table. The second question is, if the rates continue, which methods will kill all the bacteria in 320 minutes? And so if you have a bacterial count of 0 based on your trends at 320 minutes, then you want to answer that particular sterilization method. So be very, very careful. You may not have any of the sterilization methods be completely effective at 320 minutes. And if that's the case, then no method could completely kill the bacteria in that time frame. Okay, so that concludes clinical applications. Next week, I will do labs 45 and 46, and I'll also do critical thinking and clinical application for lab 25. Don't forget, lab 23 is due on Monday.